My name is Dr. Madeline Keleher, and I'm a biologist at the University of Colorado Anschutz Medical Campus. In my free time, I like hiking, cooking, and traveling all over the world. One of the projects that I study here involves obesity. So babies whose mothers were obese during pregnancy are born with more body fat, but we don't know if this is because they have more fat cells or if because they have the same number of fat cells, but those fat cells are actually larger. And this second possibility, where the fat cells are larger, has been linked to metabolic problems in adults. So we want to know if this is what's going on in kids. In the lab, I've grown mesenchymal stem cells from the donated umbilical cords of 19 babies with normal weight mothers and 17 babies whose mothers had obesity at the start of pregnancy. The hypothesis is that mesenchymal stem cells that come from the babies whose mothers were obese during pregnancy will be larger and store more fat than those that came from mothers who were normal weight during pregnancy. With a special type of food, I turned the stem cells into fat cells, and I grew them up in three-dimensional collagen matrices, which I call beads for short. In the body, our fat cells grow like spheres, but when you grow them on a traditional petri dish, they cannot grow into spheres. They kind of just grow flat in a single layer on the dish. And so that's why I'm using these three-dimensional collagen matrices, the beads, because that allows the cells to grow into spheres just like they do in the body. Once the cells are ready, I stain them with 3 colorful stains that will light up when I excite them with a laser. If you've ever had a glow stick at the 4th of July, you know that that only glows for about a day and then the glowing kind of just fades away. And the stains that I use for the cells are similar in that regard. So I end up having to do a lot of the work in the dark so that the stain doesn't fade too fast. When I've finished staining the beads, I have to be very careful when I remove the stain, especially because I do it in the dark, and I don't want to accidentally remove the bead as well as the stain, and I did do that once. I sucked up a bead and then I lost all of my cells, so now I'm extra careful. After the beads have stained long enough, they now need to be washed in order to remove any excess stains that doesn't mess with the pictures we take with the microscope. So I do 2 washes for an hour each, and when I'm done with that, it's finally ready to go image the beads. So I take them to the confocal microscope and start taking pictures. So we have the bead taken out of the tube and we've put it here onto this microscope slide, and it's in some PBS so that it doesn't dry out. We're going to take it over here to the microscope and very carefully put it down here. And then we're going to have to get the bead in focus. And so now we'll come to the computer and we will turn off the light. And we're going to actually turn on the laser. And the laser is what's going to excite the stains so that we can see it with the confocal microscope. Here we can bring up our cells. So you can see as I scroll through here, I'm going through a slice. And then we can use the joystick to move up and down so we can see different parts of the bead with our cells in it. And so now we just want to make sure that the settings are good, that we're actually getting a good image for each of them. So we just test it to make sure we're getting enough of, this is our DAPI stain. This is our Bodippy stain. It's going to stain for the neutral lipids, so you can see that there. And then this is our wheat germ stain, and this is going to show the outside of the cell membrane, so that looks really nice. So the cell that we want to take an image of is right over here, and so we're going to set the top part of the image as just above where that's going to be taken into focus, and then we're going to bring it closer into focus. So there's a nice crisp image of the cell, and then we're going to keep going down until we lose it again, and we're going to set that as the bottom part of our image, and then we're going to press go, set, and we will actually start taking the image. So the flashing that you see is the 3 different lasers. Whenever you see the blue, that's taking pictures of the nucleus with the DNA. And then where you see green, it's taking pictures of the fat that's stored in the cell. And where you see red is taking an image of the outside of the cell, or the cell membrane, which will give us an idea of the shape and the size of the cell. So this is going to go on for a few minutes, and then when it's done we can look at the final 3D version of our image. From an infant whose mother was normal weight at the start of pregnancy, and these cells are from a woman who was obese at the start of pregnancy. So you can see here that this cell is much larger than the cells from the infant whose mother was normal weight during pregnancy. And we have imaged close to 40 of these, and it turns out that the trend continues as we increase our sample size. The beads that have the cells from the mothers who were obese during pregnancy are much larger. The cells are much larger and they store more fat, so you see more of that green lipid stain.